Good evening, this is Akshara, and now I'm explaining the subtopic replication from the chapter Molecular Basis of Inheritance. So let's see. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. At the starting of the unit, they have mentioned about the scientists Watson and Crick and that they have proposed the double helical structure of the DNA. Fine, but in this particular three lines, we can expect a statement type of question. Like they can replace the word immediately and place the word lately and form a statement like Watson and Crick had lately proposed a scheme for replication of DNA after proposing the double helical structure for DNA and specific pairing scheme, which is actually false. And you can ask me why you are mentioning this immediately word so precisely. Because at the bottom they have mentioned the code that was stated by the Watson and Crick. So when you look into it, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what's this specific pairing mean? At the starting of this unit, they have mentioned that Watson and Crick have postulated the double helical structure for the DNA. Fine. After that, based on the proposition proposed by the Chekhov, they also postulated the specific pairing scheme. What's a specific pairing mean? Like the thymine pairs with adenine or adenine pairs with thymine or uracil. In the same way, the cytosine pairs with guanine. This is a specific pairing postulated by the Watson and Crick based on the proposition of the Chekhov. So first I've postulated the double helical structure of the DNA and later based on the Chekhov proposition they postulated the specific pairing scheme of the DNA, and after they've postulated the specific pairing scheme they suggested that there might be a copying mechanism of the genetic material. So here they mentioned that it has not escaped our notice. So after proposing the specific pairing mechanism scheme, they immediately got a thought that there might be a copying mechanism for the genetic material. So they immediately suggested it. So if not, other scientists would have suggested the replication theory. So it has not escaped their notice. That means they have got the thought and they immediately suggested the copying mechanism for the genetic material. So they immediately proposed a scheme for replication of the DNA. And next para, the scheme suggested that the two strands would separate and act as template for the synthesis of the new complementary strands. So if you consider, this is the parental DNA. And during the replication scheme, the parental DNA gets separated into two strands and each strand acts as a template. What does a template mean? It's nothing but a pattern that is used as a guide to make something. So here the parental DNA act as a guide to form a complementary strand of the daughter DNA. Let's know why this parental DNA strand acts as a template. We know that the DNA consists of nitrogen rings, pentose sugar, and phosphate groups. As the nitrogen rings like adenine, thymine, guanine, cytosine, and uracil follow the specific pairing rule, so here the adenine pairs with thymine and guanine pairs with cytosine. So with the use of specific pairing mechanism, they can synthesize the newly synthesized DNA strand of the daughter DNA. So they act as a guide for the formation of the nitrogen rings of the newly synthesized DNA strand of the daughter DNA. Therefore, they act as a template, and the specific pairing mechanism is useful for it. And next, when you see at the other line, after the completion of the replication, each DNA molecule could have one parental and one newly synthesized strand. So the parental DNA separated into two parental DNA strands and they act as a template and useful for the formation of the complementary DNA strand of the daughter DNA. So when you consider this two DNA, each DNA consists of one parental strand and one newly synthesized strand. And when you see the other line, this scheme was termed as semi-conservative DNA replication. What is meant by conservation? Conservation is nothing but saving and passing down. So if you consider the parental DNA, it was passed on equally to the both daughter DNA. So it is conserved but semi-conserved. So this replication can be said as semi-conservative replication. So this was actually proposed by Watson and Crick immediately after proposing the specific pairing mechanism. So we need to prove it. So the experimental proof was provided by Mathew and Franklin Stahl and later by the Taylor. So let's look into the experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plants and human cells. So we can expect a question from this statement. It was first shown in Escherichia coli. So they can change the Escherichia coli as plants, like it was first shown in plants. The DNA replicates semi-conservatively was first shown in plants. The statement is false. It was first shown in Escherichia coli. So this experiment was performed by Mathew, and Franklin Stahl in the year 1958, whereas the Watson and Crick have proposed this complementary double helical structure in March 1953, whereas the experimental proof was provided by Mathew, and Franklin Stahl in 1958. So let's look into this experiment. They grow equally in the medium containing NH4Cl where nitrogen weighs 15, and this N15 is the heavy isotope of nitrogen. It's not an radioisotope of nitrogen, it's an heavy isotope of nitrogen. Before, if you consider Hartrich's experiment, they used the radioactive sulfur and the phosphorus for stating DNA as a genetic material. But here they are not using the radioactive isotope of nitrogen, they're using heavy isotope of nitrogen. So it's better not to get confused as the only nitrogen source for many generations. So for many generations, like for synthesis of DNA, we need nitrogens because for DNA they contain the nitrogen rings. So for many generations, they provided only NH4Cl where nitrogen weighs 15 as a nitrogen source for E. coli. So what was the result? The result was that the N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. So here again you can expect a statement type of question that the N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. So the statement may be like the N15 was incorporated only into the newly synthesized DNA. No, the statement is false. Not only in newly synthesized DNA but as well as other nitrogen containing compounds as well. So the heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride. Density gradient. So if it is a radioactive isotope, we can consider it by using the radioactivity of it. But we are taking the heavy isotope, light isotope. So we can differentiate them by using the density gradient by centrifugating them in cesium chloride. Like during the centrifugation, the heavy DNA settles at the bottom and the light DNA settles at the top. So by this we can differentiate the heavy DNA and light DNA. Like heavy DNA contains N15 and light DNA contains N14. So here they have mentioned, please note that the N15 is not a radioactive isotope and it can be separated from N14 only based on densities. So let's see the experiment first and later we're gonna look into these two particular points they have mentioned. See Mathew Meselson and Franklin Stahl experiment conducted in 1958. So they have taken this DNA, which is an heavy DNA, because it contains N15, because it was grown in a medium NH4Cl where the nitrogen weighs 15, and they have taken it in a test tube and put it in a centrifugation machine, and they have got a band of heavy DNA at the bottom of the test tube because it contains N15. So later they transfer this heavy DNA into a medium containing NH4Cl, whereas here the nitrogen weighs 14. So here is the heavy DNA and they waited for 20 minutes. Why for 20 minutes? Because the E. coli takes 20 minutes to divide itself. So here the DNA replicates into two daughter DNA. But here the daughter DNAs are not heavy DNA, they're hybrid one. Because when you take this newly formed daughter DNA into a test tube and put it in a centrifugation machine, you're going to get a band of hybrid DNA in the mid of the test tube because it contains both N15 and N14. So through this we can say that the DNA replicates semi-conservatively because as Watson and Crick said, the parental DNA separate as the parental DNA strands during the process of replication, and each strand acts as template for the formation of complementary DNA strand in the daughter DNA. Look, here the parental DNA consists of N15. So this parental DNA gets separated into the template parental strand and act as template to synthesize complementary DNA strand in the daughter DNA. This newly synthesized complementary DNA strand in the daughter DNA consists of N14 because they are synthesized by using the source NH4Cl where the nitrogen weighs 14. As the parental DNA is conserved in both daughter DNAs, as the parental DNA is semi-conserved in both DNA, the DNA replicates semi-conservatively. The parental DNA contains N15 whereas daughter DNA contains N15 as well as N14 because the parental DNA already has N15 and this parental DNA strand acts as template for the synthesis of complementary DNA strand of the daughter DNA. And for the formation of this complementary DNA strand of daughter DNA needs source and the source is NH4Cl and here the nitrogen weighs 14. So through this we can say that DNA replicates semi-conservatively. And after the 40 minutes of time, this hybrid DNAs replicates again. In the same medium, NH4Cl and nitrogen weights 14. So here we get 4 daughter DNAs. So you're going to get 2 light DNA and other 2 hybrid DNA. So when you take this DNA in a test tube and place it in a centrifugation machine, you're going to get 2 strands of hybrid DNA and light DNA because the hybrid DNA consists of N15 and as well as N14 whereas light DNA consists of N14 because the only source is NH4Cl and the nitrogen weights 14 over here. So we're going to get equal amount of light DNA and hybrid DNA. The ratio is 1:1. So here you're going to get a question of what about the 80 minutes. In 80 minutes, if you consider, let's take the hybrid DNA and let's take the light DNA. And the source is NH4Cl and the nitrogen weights 14. So through this we're going to get 2 DNAs. See, one hybrid and the other light, and here both are light and here again both are light. So in this we're going to get 1 2 hybrid and 1 2 3 4 5 6 light DNA. That means the ratio is 6:2. That means 3:1. 3 light DNA and 1 hybrid DNA. So The Mathew, meselson, and Franklin Stahl proved that DNA replicates semi-conservatively. Then they transferred the cells into a medium with normal NH4Cl and took samples at various definite time intervals. As cells multiplied and extracted to the DNA that remained as double-stranded helices, the various samples were separated independently on cesium chloride gradients to measure the densities of DNA. We know that they extracted the DNA that remained as double helices because in few DNA they might be replicated completely, few may not be replicated completely, and they have extracted the DNA that have replicated completely and remained as double stranded helices. And independently, it means like independently they took the samples of heavy DNA, hybrid DNA, and also the samples of the second generation. This is the first generation, this is the second generation. So, can you recall what centrifugal force is? Centrifugal force is like apparent force that acts on the masses during the rotational motion. And think why the molecule with heavier mass density would sediment faster. Higher the mass, higher the gravitational force, and it sediment faster. The heat consists of higher the mass and gravitational force acts more on it and it sediments faster and it sediments at the bottom of the test tube. Whereas the light DNA, lesser the mass, the gravitational force acts on it is less and it sediments slower and it sediments on the top of the test tube. And next, thus the DNA that was extracted from the culture one generation, that is after 20 minutes, after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes and a hybrid or intermediate density. Why intermediate density? Because the density of the hybrid is in between the lighter DNA and the heavier DNA. The DNA extracted from the culture after the another generation, that is after 40 minutes, and second generation. So they can ask what is the ratio of the light DNA and hybrid DNA after 20 minutes, or what is the ratio of the hybrid DNA and the light DNA in the second generation. So they may ask the question using the time and or by using generation. So was composed of equal amounts of hybrid DNA and the light DNA. So in second generation, the amount of hybrid DNA and light DNA is same, that is the ratio is 1:1. If the E. coli was allowed to grow for 80 minutes, that is third generation, then what would be the proportions of light and hybrid densities DNA molecule? So light is 6 as we said and hybrid is 2, so that is 3:1. So very similar experiments involving the use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Fessia faba and faba beans by Tyler and colleagues in 1958. So first it was shown in E. coli and subsequently in higher organisms such as plants. So on E. coli, meselson and Franklin have performed the experiment on plants like Fessia faba and faba beans. It was performed by Tyler and colleagues. And before they used the heavy isotope of nitrogen, whereas here they are using the radioactive thymidine. So you need to know the difference between the two experiments. And here instead of thymidine they say as guanine, cytosine or adenine, so it's like thymidine. Okay, and next they may ask you question based on this plant Fessia faba or faba beans. They say that the Tyler and colleagues proved that DNA in chromosomes replicates semi-conservatively by using the faba beans. But you always read Fessia faba and you didn't read faba beans as it is in bracket, you neglected it. But in exam they asked faba beans instead of Fessia faba, so it takes time for you to strike the name. So while you're reading, also read the names that are present in the bracket. Read it through your mouth or while read it through your mind, but still you need to read the names that are present in the bracket so that when it comes in the exam it must not take too much of time, because even 30 seconds and 1 minute is precious during the time of examination. So the Tyler and colleagues in 58 prove that the DNA in chromosomes also replicates semi-conservatively. First it was shown in E. coli and later on plants and human cells. Thank you.